Thank you, Christophe. The floor is yours.
Thank you. Well, it's a very interesting meeting. It's also the first time I'm using this gather system, and I noticed there's the same awkwardness as in real conference about not quite knowing where to go when you want to quickly check your email. So I thought it was quite a remarkable mimicking of a real conference. So my talk, I'm going to talk about comparative genomics in particular. And well, we know we've got tons of data. We've seen some slides also to that effect already in this conference. And the question we have is, how can we devise methods that is really taking advantage of this large amount of data? And I want to talk a little bit about a framework we've pushed quite a bit in recent years. But I'd like to start just with first a question to illustrate the need for that. And so the question I have is, if you consider all of the biological annotations you have across lots of all the species that we have, which proportion do you think is based on experiments versus just computational predictions? And actually the answer to that is that it's almost everything is computationally propagated. It's around 0.1% that is based on experiment, even high throughput experiments. So, you know, our computational models are really quite important. But also what's more, if you're looking at the 0.1% and you look at the distribution of this across different species, what do you think is the proportion that is found in the 10 best studied model species? Just 10, and the proportion there is 90%. So that means almost everything we know biologically comes just from a handful of species, and everything else gets propagated and extrapolated across life. So that just shows a little bit, you know, the task at hand when we're doing comparative genomics. But of course, we can count on the fact that there's a common evolution across all of this life. And so this is going to be key to try to do this propagation. But how can we devise frameworks that could do that at scale? So this is the challenge. So it needs to be scalable and it needs to be interpretable. So if I just take a step back, we just need a few definitions for this talk. So just consider a very simple gene family. And here I have the insulin gene family. And so you might know that there's one copy in human and 2 copies in the rodents. So I've just, you know, considering here just human, mouse, and rats, and these 5 copies. And so, you know, first thing that you might do is to try to compare them among each other, and you will quickly notice that between the rodents, there's clearly like 2 pairs that are more distinctive, that are closer to one another. Kind of, you could think of these as the corresponding pairs of insulin. Whereas if you compare these now to the human, you may think, for instance, oh, let's use the rodent as a model for diabetes. Which of these 2 copies do I need to, say, downregulate if I want to have like a model for diabetes? Well, it's not so easy. In fact, you may notice actually both of them are somewhat similar. And same thing, you know, in the mouse or in the rats. So really, to make sense of this, we need to understand how, you know, what process gave rise to these 5 copies. And that's where the gene tree is a really useful concept. And so if you manage to reconstruct the correct gene tree and perhaps even manage to annotate the splits in speciation nodes and duplication nodes, it becomes quite clear that, you know, there was one ancestral insulin gene in all of these mammals, and then specifically within the rodents, you have this duplication, and it all makes sense. And it, you know, the knowledge of this tree, this model of, you know, what gave rise to these present-day genes, also gives us a way to be a bit more rigorous about what we mean about these pairs. And, you know, I'm sure that most of you know the concept of orthologs and paralogs, and so just for the sake of completeness, this is defined. And in terms of the tree, the pairs, I mean, all these pairs that I showed, actually, they're all orthologs because they are pairs of genes that start diverging through a speciation node. So for instance, the human copy with any of these rodent copies, or our two corresponding copies in the rodents. And by contrast, we've got paralogs also here. They are the genes that start diverging through duplication. So for instance, well, the two mouse copies are paralogs, or that mouse copy and that rat copy, they're paralogs. And so quite often we refer also to this structure there, which are, I mean, this graph where you have proteins or genes at the nodes and orthology relationship at the edges as the orthology graph. We'll come back to that concept. And so the framework, so yeah, gene trees, we know gene trees, orthologs and paralogs, but you can see orthologs and paralogs, so that gets quite quickly quite messy. It's very useful concept to do the inference, but then the interpretation is harder. And so the third concept I want to introduce here that is really quite central for this talk is that of hierarchical orthologous groups. And so this was proposed maybe around 2006, 2007. But I think we've really pushed this concept quite a bit in recent years, and we've been really strong proponent. We've made a number of results on this, which I'll mention afterwards. And so this hogs concept we think is really useful. And so it works like that. So it's defined in terms of the key ancestors that are in this set of species. So here we have only two ancestors really that are relevant, the common ancestor of mouse and rats and the common ancestor of human mouse and rats. And the hogs here are defined as a set of genes that have descended from a common ancestral gene in a particular common ancestor. So here, for instance, for the murine, because we have two ancestral genes, we have two hogs. Okay, and then if I go back, if I look at all the mammals, there's one gene, so there's really one hog which subsumes all of these, these two hogs. Okay, so you can see how there's, how these hogs are defined. And really, this is quite a useful way to look at the problem, because it gets rid of some of the complexity while still capturing really essential information. So why is it so useful? For the first thing is it defines quite rigorously the much more like fuzzy but still really helpful notion of a gene family and a gene subfamily. Where do you set the boundary is often quite tricky, but well, with hogs, it's very clear. So here, for instance, we will say, the family is the hog that is defined at the root of all of your species here. And so that would be this main hog, and then a subfamily, for instance, the two copies here for insulin in the rodents. Okay, so that's one thing that is nice, especially if we want to devise algorithm that then infer these families and subfamilies. We have now a definition. Second thing which is helpful is that if we're interested in particular key ancestors, as often biologists are, we can get a sense of what sort of genes they had by looking at the hogs that are defined at that level. So for instance, for the ancestral murine, we could see in this case, well, it had two insulins, and if we collect all of the hogs defined also in other families, we'll have a model of that ancestral genome. And to the extent that an ancestral genome is a bag of genes, I mean, you're going to start, maybe be able to draw some conclusions about it, what it had, what it didn't have. The third thing is, gene trees are fine for evolutionary biologists, but even for us, it can be sometimes really hard to infer and to interpret. This is just an example of a relatively small gene tree from the ensemble Compara resource. And you see MSH2 gene family. And I just want to draw your attention on the fact that there's quite a few of these nodes that are uncertain. I mean, in their own assessment, they're really not quite sure what these are. Is this speciation, duplication? Is probably a mistake in the tree. So that's quite tricky. With the hogs, we have something that is much more simple. We're not, for instance, bothering with the orders of speciations. But also from a functional point of view, it really makes sense to think of the duplication as being kind of the cornerstones, because usually we expect functional change when we have some retained duplications. For instance, these two insulin genes in the rodents, you could be pretty sure that they are doing something different. If they were kept, if they were retained in that way. In fact, that's quite well conserved among the murines. So that's probably because they do something different, and so it makes sense to have distinct hogs. that level. So, okay, I'm just going to go really quickly on this. We know orthologs are useful for lots of stuff, obviously to build gene trees, really useful also if you want to look at the history of particular gene families. Perhaps for this crowd also of particular interest is if you want to start looking at co-evolving genes, we can start looking at patterns of co-evolution, co-retention, co-duplication across time. And of course, if you're interested to do any sort of automated function annotation, you may want to use orthologs. So how do we do, so we've defined the hogs, we show it's useful, how do we infer them? So we do that mainly in OMA in the lab, and so it's a resource we've developed for many years. We start with complete genomes, and then the first thing we do is we do all versus all comparisons, so lots of alignments. To identify, essentially to try to build first this orthology graph. So this is really our first step, is what I call here the orthologous pairs. We identify these closest pairs, and then we have our orthology graph. And then from that, we infer the hogs. So we don't infer gene trees, and I will give a sense of how we can sidestep that. And then we go straight to the hogs. And once you have the hogs, you can do lots of things. You can access that in different ways. You know, just gonna mention this very briefly and rather focus on this hog inference step now. Well, actually, before I show you that, I just want to give you an example of then how the hogs might look like in OMA. So I just have here an example with this MSH2 gene, which is conserved across all eukaryotes. And so, okay, Pelagia is really quite small here, but this is a tree of all the eukaryotes in OMA. So you can see here, we have actually here the primates, and we've got vertebrates here, we've got plants, fungi, some protists here. The protostomes are here, you see, Drosophila. And so if you go in OMA and then you look at, for instance, this MSH2 family, you will see, you know, if you zoom in the primates, we have a excerpt of the species tree here, and you can see MSH2 has been classified in that hog here. Each square is a gene, and we have this bar that separates the hogs defined with a particular common ancestor. So at the level of the, you know, the common ancestor of all the primates, we think there was one MSH2 copy and a few other paralogs. And, you know, maybe it was lost in a few species, maybe that these are annotation errors. But, you know, so it's classified in terms of the primates, and you could go deeper in the tree. You can look, you know, here we're going to the fungi, and because it's really the result of some really deep duplication, we still have these 4 copies, which we can relate. Okay, you know, in some species it's duplicated. Maybe it's a sort of a hybrid here. Some have lost. You can look at the pattern. And here we are all the way to the plants. The plants have another 2 copies. But you can see this is an example where it's really quite nice. We could relate all of these genes here, thousands of them across all of eukaryotes in a way that is really fairly scalable. I hope you'll agree with this. Certainly easier to interpret than you know, like say a gene tree now with all of with let's say 2,000 or 3,000 tips. Okay, so how to do the inference? I just want to give you a little flavor of that. So maybe one key result we contributed a few years ago is we noticed actually if your orthology graph is perfect, there is a bijection between subgraphs that are defined at particular for particular set of species and the hogs defined for the species. So just a bit slightly more formal, but you know, it's really the same thing as I just mentioned. If you take the orthology graph, it's complete, it's perfect. And we now restrict this to a particular class of species. We get a subgraph. There's a one-to-one correspondence with the hogs defined at that level. And so that's very nice because it means we can skip the tree inference process, which is so tricky. Provided we have a perfect orthology graph. Now, of course, it's not perfect, so we have some heuristics to try to do a reasonable job there with the inference. And these algorithms have been refined a little bit, and there are some reference in case you're interested. So in particular, one that we published a few years ago that is much faster and that essentially infers all the hogs in OMAF, starting from the orthology graph in just maybe less than a day, without even parallelism. Once we have done we've created the orthology graph, which contains, by the way, hundreds of billions of edges. So it's not a trivial graph, but it's manageable because we can slice it in subgraph restricted to particular clade of species. And then once we have the hogs, we could do other things on them. We can use this as to relate, maybe, for instance, in this case, some functional information across different species. So here is QTL experiments, so association between particular traits and genotypes. And so usually this only gives us a bit of a window around the genome where this is where there is maybe the causal genes, and we can use the hogs to integrate different QTL experiments across different species. We've done some software to visualize and analyze hogs because it's a bit of an emerging kind of data structure. And more recently, we published some work to identify co-evolving hogs. And so that's in that reference, if you're interested. And maybe just something I'm going to mention in the next slide we're quite excited about is once we've done these hogs, which are really kind of model of the ancestral genes across all of these species of interest, maybe there's an interest now in placing new sequences within these hogs really quickly, really efficiently. A little bit like, in metagenomics, people are sometimes trying to do some placement within known taxa. Here it's a little bit more subtle because we want to place genes into the right family or the right subfamily, right? The right hog. And actually, when we started working on this, we realized it's really not as simple as we thought. I mean, maybe naively you might think, okay, why don't I just do a blast and assign my sequence to the subfamily of the closest or the top hit? If I get, for instance, here a new gorilla hemoglobin, well, the globin family has duplicated at the bases of vertebrates into myoglobin and hemoglobin. So myoglobin that carries oxygen in the muscle and hemoglobin in the blood. And so, you have a copy of that gene from the gorilla. It's going to look very much like a hemoglobin from the chimp. And so, yeah, sure enough, it's also hemoglobin. You're all good. But consider this example. Let's say here you take a sea urchin globin here, right? We are before the duplication. Well, the closest sequence, even if your analysis is perfect, is going to identify here, still again, this chimp sequence as the closest one on the tree. And so you're going to misclassify it. So the closest sequence is not necessarily in the same subfamily. In fact, we did some comparison where we used a different number of different approaches to find the closest sequence. For instance, Smith-Waterman, but also much faster heuristic like Diamond. And you could see that in terms of placing in the right family or subfamily, well, you don't really have a control over precision and recall. You cannot really trade off. You're always going to pick, the family of the closest sequence, which is maybe not too bad for recall, but you make quite a few mistakes. And if you take care of that problem, you can do much better. So you see, for instance, here, I'll just give you an example with Amborella. This is a sister species to all of the flowering plants. So it's a bit more of a more tricky case. And you can see, you do a lot of mistake. Only about half of the genes are placed in the correct family if you just take the closest sequence because of this type of problems. And if, with our approach, we can fix this essentially. It's not going to be perfect, but the precision is much higher, and we can trade these off. And another benefit of that is we could be much faster now, because since we're kind of indexing at the family level rather than at the sequence level, as you increase the amount of data, the number of families are not going to increase quite as quickly. And so the performance is I think much nicer. So I just want to finish with one application just to show you then what could we do if once we have the hogs concretely. So that's just a paper that just came out last week in GBE. There is collaborators who are interested in parasitism, well, the emergence of parasitism in trematodes. So, you know, they have typical, like lots of genomes, and they want to know, okay, we think around 400 million years ago, these clades, you know, became parasites. And so, you know, is there any evidence of that in the genome, and, you know, what might have happened? So again, you know, we can reconstruct the hogs for that branch, for the last common ancestor, get about 13,000 or so hogs, so, you know, suggestive that maybe we had around that many ancestral genes. But then also we could do that at every node and then keep track of the changes on these branches. And we might see that, well, actually, you know, they've lost some genes, some were retained, some duplicated by keeping track of the hogs defined at each level. And just a fun thing here about the duplicated genes here, if you look at the most duplicated ones, the function they have seem to be quite plausible for the emergence of parasitism, you know, things that have been otherwise characterized as you know, being associated with behavioral, host behavior manipulation, hiding from the host immunity, etc. So I'm just running out of time, so I'll just conclude. So I think, you know, the hogs framework is really a scalable way to integrate information from lots of genomes. And in particular, it reconstructs these key ancestors that we can hope are getting increasingly accurate as we add more and more data, which is kind of in contrast to the typical comparative genomics approach where you know, I don't know, you start maybe with one-to-one orthologs, and then as you add more and more genomes, your set of one-to-one orthologs shrinks and shrinks and shrinks, and then you have some trouble, or you do pairwise comparison, but then how do you integrate all of this information? And then finally, specifically, but that, you know, I mentioned also some other application. You could study gains, duplication, losses in particular parts of the tree of life. So thank you very much. These are just the people who were involved in this work, the funders. And again, thank you very much for this nice meeting.
Okay, so thank you very much.
Over time.
Sorry.
Yeah.
Okay, thank you very much, Christophe, for this great talk. So now we welcome questions. So you can either type them or you're welcome to speak up. I see that there is already a question in the chat. The question is the species tree fixed to infer the orthology graph?
Okay, that's a very good question. So with the hogs framework and the algorithm I mentioned, we need to fix the species tree. Now, how do we get the species tree could be like a very contentious now discussion, but we use orthologous markers that are obtained in a different way without any assumption about the species tree. But it's kind of two complementary process. One is to get a good species tree, and for that we can be a bit choosy in which genes we want to consider. And sometimes we just use kind of some consensus from the literature or the NCBI taxonomy. The species tree does not need to be perfectly resolved for our application, because as you could see, we don't try to necessarily resolve the speciation nodes. On the other hand, once we have that, then we do all of the hogs analysis, which is more of a kind of a gene family type of analysis. But that's a very good question. I hope I answered it.
Okay, thanks. I mean, Louisa, if you would like to add to your question, feel free to do so.
Yeah, thanks for the nice talk and the answer. I think you answered, but I'm still thinking for certain.
Sorry, your voice is a bit low. Could you please increase?
Get closer
Can
to
you
the
hear?
mic.
Yes, much better.
It's better?
Yes.
Okay.
Thank you.
Good.
Yeah, thanks for the talk and for the answer. I'm just thinking for some parts of the tree that are not so resolved, and then you might assume some gene history depending on the fixed species tree.
So yeah, again, I think so all I told you is like given a particular phylogeny, but it can be unresolved in the sense that we we, for instance, I mean, we could go in, you may be thinking about issues of incomplete lineage sorting. I mean, for some of those of you who are familiar with this idea, the order, the branching order of the gene tree may not even be consistent across all of the families, if you even absent any duplication. But that is somewhat abstracted from the fact that we don't try to resolve this speciation. In fact, that our inference is more robust than if you constrain yourself to reconstructing this history. And so where we have a limitation is if you have a duplication that is shared by a subset of these species that are attached to a multifurcation, there, it's not going to be very clean. It's perhaps, we will infer then a duplication on two branches, on multiple branches. But by and large, the number of really true duplication is not that high compared to, let's say, a sampling that is going to increase and get denser and denser and denser. So we're quite happy with even having like unresolved part of the tree. And maybe, in some case, we have like, it looks like two independent duplication, but it's not. Or it looks like a duplication that happened in the root of this whole set and then was lost in one of them. But it's a bit like a local mistake that doesn't have like quite the same consequences as if you get your gene tree wrong and you start doing your reconciliation, at face value.
Okay, thank you very much.
Thanks. I see we have a next question from Maxime Godefroy. He's asking, would it make sense or would it be possible to infer the orthology graph from a set of gene trees?
So you could, but the problem is just what is easier and what gives you the best result. And in practice, when people are building gene trees, they usually start with some sort of pairwise clustering approach, which tries to wind things down a little bit, because some families are just huge. If you take some, some extreme example are like olfactory receptors, where you have maybe 1,000 copies in a single genome. So now, how are you going to build a gene tree that contains potentially millions of taxa? So, but on the other hand, there are methods that do that, that are tree first, and then once you've got the tree, you've got everything. You've got the orthologs, you've got the graph, you've got also branch length. In a way, you could take any projection that you want then from this tree. But in our experience, it's not so easy. Just think also about the issue about rooting these trees. You don't really have an outgroup the same way you have even with species. So you can, if you're good at building gene trees, you could do that. Yeah.
Okay, thanks. Maxime, if you want to add to your question, feel free to intervene now. I mean, if not, we can continue. Yeah, maybe, since we are talking about these gene trees versus species trees, etc. So if you were to consider your method in some bacterial species, can you find out about horizontal gene transfer, for instance?
So that's, okay, very good question. So in our model of the hogs and this correspondence and so on, we don't really model horizontal gene transfer. However, we found that it's not hugely disruptive. I mean, what happens is that okay, you're going to have like some groups all of a sudden that require a lot of many losses to explain that, and it's probably it's not going to be really plausible. But more locally, it won't disturb so much. But the other thing is, and maybe I'm already anticipating a little bit Eric's talk later in the week, but actually, the evolution of bacteria, I mean, okay, it's not tree-like in a conventional sense, but there is actually, the species tree is not such a bad guide to deciding which order you want to group things, actually, for a reason that will become apparent later this week. And I should also say there are also quite a bit of vertical inheritance in bacteria too. So we see lots of duplication. I think they tend to be ignored. People just think presence, absence. But actually, some gene family, it's quite clear there's multiple duplication and then retention of this duplication, which makes us think, okay, it's not just like going all over the place like crazy.
Okay, well, thank you very much. So yeah, given the time, I think we should thank again Christophe for his great talk. And now we welcome our next speaker. So our next speaker is Tandy Wano, who is on the faculty